Hormones of the endocrine system function to alter the activity of particular cell types. And they do this by altering the types, amounts, or activities of particular cellular proteins. So we need to think of our cellular proteins as the structural and functional elements of the cell. And by modifying those structural and or functional elements, we actually change the phenotype, the expression of that cell, what it does, what it looks like. Right? And so hormones, the classic definition of a hormone is a blood-borne chemical messenger. So let's draw a capillary off to the side of this in this tissue. Snipped off a little bit here. And what would happen is our blood-borne chemical messengers can diffuse, right? They've gotten into the bloodstream from their endocrine glands or endocrine tissues, right, where they're produced, and then they travel around and around within the bloodstream and can readily diffuse out, often readily diffuse out of those capillaries and into the interstitium, right? So we have these guys entering the interstitium of this particular tissue via diffusion, right? And then they can modify that cell's function. And they're going to modify it based largely upon the chemical class of that hormone, whether the hormone is hydrophilic or lipophilic. Right? Another possibility for a chemical agent acting in this particular fashion would be to have an adjacent cell producing these chemical agents, releasing them into the interstitium. Right? So these are not going to be blood-borne. So in this case, acting in very similar fashion, but in this case, we're going to call it paracrine as opposed to endocrine, because these are cells that are located locally adjacent to this target cell. Now, in either case, lipophilic hormones or paracrine agents can readily diffuse through the lipid bilayer, right? So we can pass through this membrane to bind receptors that are found in the nucleus or sometimes in the cytoplasm. And they tend to alter the types or amounts of cellular proteins, right? And so if we make new proteins, what we're doing is we're making proteins that are not currently expressed by this target cell. If we make more of a particular protein that's already being made, we also change the cell's function. Alternatively, hydrophilic hormones can't enter the cell and would have to bind to a membrane-bound receptor with an active site that faces the interstitium. It's important to note that it binds to this active site randomly via diffusion. But once bound, we activate an intracellular mechanism that leads to activation of inactive proteins. So in this case, we're altering the functionality of proteins that already exist, right? So as I said, we're altering the types, new, amounts, more or less, or activities, right? Making an inactive protein active of cellular proteins to change their function.